Yeah, so this is, New York genomics researchers being overwhelmed by the great wave of genomic data. So, next slide, okay. So over the course of about 15 years, the genomic landscape of major cancers was defined by a series of projects listed here and others. I had the pleasure of being part of the Cancer Genome Atlas. It was very exciting to, see the full complement of genomic alterations in these major common cancers. On the clinical side, the year that really changed everything for solid tumor oncology was 2004. Three groups independently discovered EGFR mutations in lung cancer. Actually, Matt Myerson's group, with the help of the Japanese collaborators, and a group at MSK as well, led by William Powell and Harold Varmus. So at that point, there was one biomarker that was linked to targeted therapy. But as we all know, there's been an explosion of targeted therapies linked to specific biomarkers. This is shown here, a recent review in Cancer Discovery by Sarah Swenholz and et al. From our center. So you can see EGFR in 2004, and then this huge growth in FDA approvals linked to specific biomarkers. For instance, even going from 2017 to September 2023. So we're faced with having to characterize so many different biomarkers at the same time, so to speak, that the most efficient way, of course, is through large panel sequencing. So if you think about the cancer workup, the old cancer workup, 20th century, really you needed to know two pieces of information: the type of cancer and how far has the cancer spread. I would posit that in the 21st century, there's a third piece of information that we need to know: is what genetic alterations drive that cancer. So our molecular testing strategy at MSK, we have a rapid panel for common critical targets. I won't be talking about that. We have comprehensive NGS-based assays to identify driver genetic alterations in all solid cancers. There's MSK Impact, which is our DNA-seq panel. MSK Fusion is a targeted RNA-seq panel, and MSK Access is a liquid biopsy panel. And each also has a version for hematologic malignancies. I will actually mostly talk about MSK Impact data. So, the original goal of MSK Impact was to efficiently identify biomarkers linked to targeted therapies. But what we've discovered is that this kind of program has many additional benefits beyond the identification of therapeutic targets. It really provides a more complete picture of each patient's tumor, the predisposing germline variants that patient carries, and the state of hematopoietic aging as indicated by clonal hematopoiesis. It also enables now, as opposed to single gene testing, it enables combinatorial targeting, either pharmacologic or immuno-oncologic. So this is MSK Impact. This was an assay that was, the prototype was developed by my colleague Mike Berger in his lab, and we moved it into the clinical lab, into clinical production. Some of the dates are listed there. We actually submitted it to New York State Department of Health for review and approval. Every clinical laboratory assay in the state has to undergo review by the New York State Department of Health. We got the approval in 2014 and initiated testing. So the assay is a matched tumor normal assay, and it's specifically designed to work well with formalin-fixed paraffin-embedded tumor. In fact, if we get frozen tumor, it's like, what do we do? The technologist said, what do we do with this? And normal blood is usually the matched normal. It's gone through different versions. We're currently at 505 genes. It's a hybrid capture assay, and NovaSeq sequencing, and actually, thank you, Nicolas, for having gone through an example of a bioinformatics pipeline. The one we use is probably very similar. We pick up all the major types of genomic alterations, including select rearrangements, and these are some of the papers. There's actually a paper on the first 50,000 cases that's about to be submitted. So the panel is carefully selected genes that are implicated in cancer through DNA alterations. So all the protein coding exons of 505 genes, some introns of genes that are commonly rearranged in cancers. Because it's a hybrid capture assay, we can detect rearrangements using that approach in some instances, as well as additional probes for copy number and QC checks. So one important aspect is that, especially initially, this was being used in patients with advanced cancer who often do not undergo resection. They get a biopsy, sometimes they only get a cytology. So we needed an assay that worked on those kinds of samples, and indeed this assay works quite well on small biopsies, cytology, and of course it works on resections. So again, original goal was to more efficiently select patients for targeted therapies. The poster child, the prime example of this is lung adenocarcinoma. At least 50% of patients here, when studied by MSK-IMPACT, have an alteration that corresponds to an FDA-approved therapy. And the proportion, certainly, of EGFR in Asia is obviously higher. So even more reason to use this kind of approach. Just a brief word on complementary targeted RNA sequencing. Not all fusions can be detected by genomic DNA capture-based methods because some introns that are involved in rearrangements are huge, and you would eat up too much sequencing capacity. So the solution is a targeted RNA-seq assay based on the Archer technology that we've called MSK Fusion. And this was a paper showing how applying this to cases that had no driver, and by driver here we mean driver that activates the MAP kinase pathway. So no MAP kinase driver by MSK-IMPACT. They underwent targeted RNA-seq, and a good number contained fusions, and some of these, the patients actually were able to benefit from that additional analysis. So it's really, MSK-IMPACT is the centerpiece of our institutional clinical genomics initiative. which includes a patient consent, of course, for various reasons. But one of the benefits is that the consent also allows us to do additional studies with the results of the sequencing and with the leftover DNA. All the cases are reviewed first by a bioinformatician and then by a molecular pathologist and molecular pathology fellow. And then the question of interpreting the variants comes up. MSK has created this effort called OncoKB, which is a precision oncology knowledge base that is curated basically by a team of experts at MSK and is updated, is continually being updated, such that clinical reports are automatically annotated with the latest interpretations. This is the OncoKB website. It's led by Devyani Chakravarti, and it's free. It's open to for nonprofit institutions. It's entirely free, and it has really detailed information. Of course, it's U.S. based, so the levels are defined by FDA approvals and NCCN guidelines. So, like I said, the original goal was to streamline patient selection for targeted therapies. We so far have sequenced with all of those assays shown here over 122,000 samples from over 86,000 patients. But there are three aspects that have really enabled our clinical genomics initiative to go far beyond the original goal and really serve as a nidus for additional discoveries. Number one, is a systematic, almost immediate data sharing within the institution. So we review cases during the day, and that evening, the results of that case are de-identified and are transferred into the CBio portal. Anybody in the institution can see the results. All the researchers, the basic researchers, clinical researchers, fellows, and so on. And this has resulted in an enormous number of studies and publications. We also shared the data with AACR Genie with a 1-year delay. But I don't want to brag, but I think we are still the biggest contributors to AACR Genie. And, well, we'll leave it at that. The other important, absolutely critical aspect that distinguishes our assay from, for instance, Foundation 1 is paired tumor normal sequencing, and I'll get back to that. And then we're finally we have such a big data set at this point that there are these emergent properties, co-mutation patterns, or rare but recurrent hotspots. So this is one way to kind of look at the different types of information that we're getting. So we sequence tumor normal. Variants that are only present in the tumor but not the normal are somatic variants. Variants that are present in both tumor and normal are germline variants. But then there are variants that are only present in the normal DNA from the blood, but not in the tumor. Well, actually, they're present at a higher level in the blood than in the tumor, often. And these are clonal hematopoiesis variants. Clonal hematopoiesis has been known for many years, but never before have we routinely seen it. I mean, routinely identified it in patients. And this has led to some interesting studies. The other interesting aspect of having tumor normal is that there's also a whole, we report the alterations listed here in the clinical report. There's a whole set of other types of analyses that are investigational, that are not part of the standard report, that are of great interest. So having tumor normal actually made it very easy to take this algorithm called MSI sensor that had been developed at Washington University by the group led by Ladanyi for use on paired tumor normal exomes. We adapted it for our tumor normal panel data, and we're able to call microsatellite instability. So here's an example of a stable locus and an unstable locus, and it turns out that the panel itself, even though we didn't specifically design it at the time, contains over 1,500 microsatellite loci. So now you're getting information on microsatellite instability in every single cancer. Whereas typically, historically, only colorectal cancer and endometrial cancer were studied. And you can see that here, where an MSI sensor score greater than 10 indicates instability. As this was thoroughly validated by orthogonal methods, and you can see that colorectal and endometrial are at the top, but there are all these other patients in this red square, and it corresponds to 39% of all patients with microsatellite instability are not colorectal or endometrial. As we know now, these patients are candidates for immunotherapy and would have been missed historically, but now that we have this information routinely, this has become very useful. So the matched normal sequencing also can help with cancer risk assessment, both because of the germline analysis for cancer predisposition genes, but also because of the detection of clonal hematopoiesis. So clonal hematopoiesis, like I said, is something that hadn't been tested for routinely across large cohorts. And in studies led by our former lead clinical bioinformatician, Ahmed Zahir, who unfortunately left, led to a series of papers based on the MSK impact data. I don't have really time to go through these papers in detail, just a couple of little highlights. So the prevalence of clonal hematopoiesis is proportional to age. It's an age-related alteration. And we detected in about 25% of solid tumor patients. This includes, obviously, most of our solid tumor patients are older adults. And in 4.5%, the alterations are presumptive leukemia driver mutations. So you see here DNMT3A, TET2, ASXL1, and so on. And for patients who do have presumptive leukemia driver mutations, there is a risk of developing leukemia. I should point out that the y-axis here is from 0 to 3% at 18 months. A practical point that is very important is that if you only sequence the tumor, you will misinterpret some clonal hematopoiesis variants because tumors often usually contain white blood cells. And if the white blood cells contain a CH variant at a high variant allele frequency, that will be picked up and interpreted as coming from the tumor. And this phenomenon actually becomes worse and worse with age, as you would expect. So if you're, sequencing the tumors of individuals over the age of 70 without the matched normal, you run a high risk of overcalling mutations in the tumor. And these are the mutations that typically get overcalled. So I think we already know that if we see a DNMT3A mutation in a solid tumor, we kind of suspect it's not coming from the carcinoma cells, it's coming from blood cells. But there are other mutations that could be either, obviously, P53, SF3B1, CHEK2-ATM, and less commonly, but really problematically, this also happens for KRAS, NRAS, and IDH mutations. So KRAS and NRAS mutations can occur as CH mutations and be misinterpreted as coming from the tumor with clinical consequences. Now, for variants that are present in both blood and tumor, these are the germline variants. It turns out that all the major cancer predisposition genes were already in the impact panel, even though it wasn't designed on purpose like that. So patients now have to opt out of germline testing. So there's an opt-out consent. And if germline testing is initiated, there's a separate pipeline for variant calling in 90 cancer predisposition genes. The remainder of the genes in the panel are not called for germline variants. And this was confirmed to be very effective, was validated as a diagnostic panel for germline cancer predisposition variants. And it has really changed the paradigm of germline clinical testing. So originally, historically, germline testing for cancer predisposition was based on guidelines, like colorectal cancer under 40, family history, et cetera. And the patient was referred to a clinical genetics service, and eventually, if they agreed, then the patient was sent for testing, et cetera. Now, it's a new paradigm. The oncologist offers germline testing as part of IMPACT. Consent is obtained, and then the analysis of 90 cancer predisposition genes, and the results are returned to the patient and the oncologist with notification to the clinical genetics service. And the experience with this was reviewed, the initial experience was reviewed back in 2017 by Diana Mandelker from our team, the first 1,000 patients who got this kind of testing. So again, these are patients, all comers. There's no selection for family history. 17.5% had a pathogenic germline variant linked to cancer. And of these, 55% would not have been tested based on clinical guidelines. So this is, this is a finding that's been reproduced elsewhere. There's always about 10 to 20% of patients of adults with solid tumors, no obvious familial history, who have a germline variant that is informative. Now, one interesting thing that we've done is that has been made possible is tumor type prediction based on the genomics. So a couple of years ago, again, my colleague Mike Berger Took about 8,000 cases that have been sequenced across 22 cancer types and used the random forest classifier using these types of alterations to predict the cancer type. And this is something that experienced molecular pathologists might be doing subconsciously, but here it was done very systematically. More recently, and this just came out, is an update of this model that's now called GDDENS, that was trained on substantially more data, more cancer types, more genomic features, and that used a neural network instead of random forest. I couldn't tell you the pros and cons. Maybe Nicola can tell us. Yeah, these are the genomic features that were entered into the neural network. So there's a prediction made, a percent probability of different cancer types, and you can see which features of that patient sample are driving that classification. So the specific features are most of them are familiar to us. So these are the features that the neural network uses to identify the cancer type. So for colorectal cancer, APC, of course, KRAS, other alterations that may be less familiar to if you're not into colorectal cancer, but 20q gains, SOX9 mutations. Anyway, all these point to colorectal cancer. Conversely, cutaneous squamous cell carcinoma, the strongest element in the prediction is the UV signature, and then other mutations such as TERT promoter and so on. And so this is actually, when we're reviewing these cases, we now see the tumor type predictor. We don't report it clinically, but it is helpful in sometimes interpreting the genomic data. Sometimes there's a situation where the pathologic diagnosis is carcinoma of unknown primary or poorly differentiated carcinoma, and this can really clinch the diagnosis. So in this case, we see what was driving the assignment of a 99% probability of colorectal cancer. So again, APC, KRAS, 20q gain, etc. Finally, in the final part, I want to talk about how this clinical cancer genomics is enabling combinatorial therapeutic approaches. So the irony is we study 505 cancer genes, but the oncologist only treats one oncogenic alteration at a time. And if you look at this patient's report, you can see that there are actually multiple targetable alterations, including EGFR, ERBB2, and actually now MDM2 amplification is also targetable, as I'll get to. The problem is TKI, treating one oncogenic alteration at a time with a kinase inhibitor inevitably leads to the acquisition of resistance mechanisms. My group has actually defined so many of these mechanisms that it's a little bit discouraging, to put it mildly. It makes for a lot of papers, but ultimately it points to the not the futility, but the limitations of monotherapy. So this is how I portray the oncologist fighting the medusa of TKI resistance mechanism detected by the molecular pathologist. If we think of the analogy of AIDS, it wasn't until triple therapy was introduced that AIDS really became a chronic disease that was under control. And so the question is, why can't we do the same in cancer? This is the paper I referred to earlier from Sarah Swenholz and Debiani Chakravarti from our group that quantifies the increase in actionability in patients with cancer. So they noted that between 2017 and 2022, which is just 5 years, it went from 10% to 31.6, 9% to 31.6% of patients that had an actionable genetic alteration in their tumor. And what's more interesting from my point of view personally is they also did an analysis of the number of actionable alterations per patient. And they, at my request, they actually did an additional analysis which excluded MSI high and TMB high cases. So these are all kind of classic targetable genetic alterations. And you can see the proportion that have 2 or more actionable alterations for different types of cancers. So for many cancers, almost 50% have 2 or more actionable alterations. So there is this opportunity for combinatorial therapy. So we were interested in this. One thing that caught my eye early on is this unusual association of MDM2 amplification with certain TKI-driven subsets, RTK-driven subsets of lung cancer. So there's this very strong and unexplained association with MET exon 14 skipping alterations and RET rearrangements. There's a weaker association with EGFR mutation, and there's a negative association with KRAS mutation. So we wanted to see if combining MDM2 inhibition with either inhibition of the activated RTK or inhibition of the MAP kinase pathway downstream using a MEK inhibitor would work better. And indeed it does. So these are, I'm skipping all the in vitro data. So these are patient-derived xenografts representing 3 different kinase-driven lung cancers, EGFR, MET exon 14, and RET fusion, each one with MDM2 amplification. I should say, as background, MDM2 amplification is mutually exclusive with P53 mutation because they're functionally redundant. So these were all P53 wild type. And the combination in every patient was superior, sometimes substantially superior to the individual drugs. And this was published last year and is now being translated into a phase 1/2 clinical trial for patients who have progressed on standard RTK inhibitors led by Greg Riley at our institution. But the question is, would it work even better if it was given in the first line? So yes. So we did that again with PDXs. And here, 2 different PDXs, a RET fusion and EGFR-driven PDX, both MDM2 amplified, P53 wild type. And here we actually used two different MDM2 inhibitors. And you can see that there's virtually no growth of the PDX tumor way out, 200 days here. It was 85 days here. In this case, at 200 days, there was no measurable tumor, and this was close to unmeasurable with the other MDM2 inhibitor. So if we could convince the companies to run a clinical trial, that would be very interesting. But unfortunately, the problem with clinical trials is each company has one drug, and they don't want to talk to the other company and have a combined clinical trial. So it makes this kind of approach difficult. There's also, I'll end with this, the clinical genomics also offers an opportunity for combinatorial, personalized immuno-oncologic approaches. So interestingly, what were these two doing before COVID? Well, this was like the last review article they wrote before COVID, and they were proposing a personalized vaccines for cancer immunotherapy using RNA vaccines that could be rapidly produced from the mutations in a patient's tumor. And this turned into a trial. So here they are again, Sahin and Turechi. A trial that was led by Vinod Balachandran at our institution that actually occurred during COVID, and that tested this concept. And so the patient's tumor was sequenced, the mutations were identified, there was a selection of the top 20 neoantigens, predicted neoantigens. A custom mRNA vaccine was manufactured and was administered according to this schedule to patients with pancreatic cancer who also got the FOLFIRINOX standard therapy. And so this is obviously a pilot study, but in patients who showed evidence of response to the vaccine, there was a 100% survival at 30 months, which is pretty impressive for pancreatic cancer. And perhaps not surprisingly, patients who did not show an immunologic response did not do very well, had a more typical course for pancreatic cancer. But nonetheless, very promising, and a much larger trial is in progress, led by Vinod Balachandran. The same approach can also be used with peptide-based vaccines. So this was a trial of using basically the same concept in lung cancer. So identifying the mutations and then creating a peptide vaccine, a vaccine with up to 20 peptides representing neoantigens in that specific patient. And this was led by Marc from Harvard, from Dana-Farber, and just shows, again, the potential, of how a deeper understanding of a patient's tumor can create new opportunities for personalized targeted oncology approaches. So I'm going to stop there. And thank you for your patience and attention.
Great talk. Thank you, Dr. Ladanyi. I think we have time to take a couple of questions.
Yeah, thank you very much. It's very interesting and very suggestive presentations. I have one question about your co-mutation analysis of your large impact data set. So you see some coexistence of driver mutation in the single cases. And have you ever checked these 2 or 3 mutations occur the same cell? Or, same tumor cells. So you did some single cell analysis or some fish or some kind of molecular technique to see that these co-mutation occurs the same cell.
Yeah, that's a great question. The vast majority of studies at diagnosis show that they're in every cell in the same cells. In samples from patients who've progressed on targeted therapies, there can be this heterogeneity of alterations. Some cells have one resistance mechanism, another subclone may have another resistance mechanism. And that's for mutations. For gene amplification, in a way, it's much easier for heterogeneity to occur if, by chance, one cell gets more of the extrachromosomal DNA fragments than the other in a given division, and so on. So that in that context, in gene amplification, there's more heterogeneity.
Thank you.
So about the classifier of the different tumor type, isn't it more interesting to look at the case where it fails, where it doesn't classify correctly, and to tease out the reasons for that? Or, it's not more interesting, but also interesting?
Well, it's the usual answer is that you need a minimum number of each cancer type to train the classifier. So they trained it on 38 cancer types, but there are many more. So the typical reason it doesn't work, the kind of trivial reason it doesn't work, is that it's a cancer type that was not originally in the classifier. I think what you bring up is, let's say we have a classic colorectal cancer, but it's missing, for instance, APC mutation. That might trigger RNA-seq study because there are these fusions, R-spondin fusions, that can substitute for APC mutations, but we don't pick them up on impact. So it may trigger additional studies.
Yeah. And as you can expect, I will say must trigger whole genome sequencing.
Yes, ultimately, that would be the simplest, yes.
Anyone else?
If Yoshiko has a question, and you can ask a question.
Finally, I get to ask. So I have a quick question. It was great to see how such a through large-scale study led to those many insights by sequencing 505 genes. I feel like at some point, do you feel like we have enough information, so let's reduce the number of the genes? Do you think that they will come, or do you think we're going to keep sequencing? And the other thing I was thinking was probably insurance payers' pressure. Sometimes they would like to sequence fewer number of the genes instead of being very thorough. I would love to hear your opinion on that.
Well, start with the second one. I think since you work in the U.S. system as well, it's discussing insurance, billing and reimbursement is a whole other, meeting. Would take a whole other meeting. In terms of the size of the panel, it turns out that it has been very beneficial because with small panels, you cannot estimate tumor mutation burden accurately. With large panels, such as ours, the tumor mutation burden is accurately measured. So I don't see us reducing the size of the panel. In each version, we've had a discussion of, let's see the genes that were never altered. Maybe we should remove them. But ultimately, we decided not to, so it's incremental. At the same time, we have a paper about to be submitted where we did whole exome sequencing in cases that had undergone impact, and the yield of additional like driver mutations was very small, very, very small. So I think we have a good panel. We certainly don't have every cancer gene that will ever be discovered, but I think it's a very good panel.
It's probably a good range right now.
Yeah.
Great, thank you.
Any questions? Yes, go ahead.
So recently, so many people interested in RNA splicing alterations. So your RNA panel can detect such kind of splicing alteration or variants, or?
Right, but it's a targeted panel, so we can detect med exon 14, the splicing variant. We can detect the androgen receptor V7 variant. Trying to think of other, the EGFR V3 variant that's seen in gliomas, but it has to be targeted. So it's not a pan.
You captured you capture exon, or you did just a single?
Yeah, the targeted
capture
RNA-seq is based on anchored PCR. So it's one gene-specific PCR and one adapter PCR. So primary, I should say.
Thank you.
Thank you for very comprehensive. It's such an amazing system. I wish I could go to MSK when something happened to me. But anyway, could you update the cancer vaccine part, because it has a long history. It's been a long time, but haven't heard these days. So if you could update about it.
Update.
The current situation, it's kind of coming to close. So it's developing, or someone is not pursuing, or that kind of. So is it going strong, the cancer vaccine?
Oh yeah, I mean
Yeah.
there's an enormous push in immuno-oncology at our institution. Every type of approach, CAR T cells, vaccines, obviously checkpoint inhibitors. Yeah, it's.
Yeah, I mean, sorry, so the mRNA vaccine, what I meant was the.
Yeah.
Yeah,
We're
okay.
There are different trials in different stages of development that would use RNA vaccines.
But what's novel is that it's personalized.
Yeah.
That's so it exists before, but where you would take the most common variants and build a vaccine for everybody. Now with those approach, you sequence the tumor and you build a vaccine for that particular patient, right? And so there will be overlap between the patients, but the vaccine is really for that particular patient with those HLA allele in that patient, and you build something for that one individual.
That's going to be very complicated because you find the neoantigen first and then all the HLA sequencing and all the...
I forgot to mention, Impact also gives us the HLA alleles.
Yeah.
So that information is available at the same time. But on the other hand, I would say if you think that's complicated, CAR T cells, now that's complicated. Anyway.
Thank you.
So I do not know the details of the techniques, but from my understanding from your lecture, I think the technology is really advancing in the log magnitude. So probably you can do the same thing in 1/10 of the time. So probably each sequence needed months and years to make it happen, but now probably it can happen in 1 week or 2 weeks, and probably that's one of the reasons the concept existed before, but it's happening really in a high specific methodology. And Mike, I do have one question. It's regarding the prediction of future cancer risk. If you check the cancer gene or you have potential positive, and if you do that on everybody, nearly 1 out of 5 people has something scary. So no, let's say if I do that on all patients of mine, 1 out of 5 people come in. I'm going to have cancer.
All your cancer patients.
So basically, then how can I? So if that is already happening in commercialized cancer, gene analysis or something. If you send the cell from the mouth, and they tell you, oh, you are dangerous, and they are blasted with so many dangerous information these days. So how is there any way that you can tell the patient don't panic?
That is the specialty of our clinical genetics clinicians and genetic counselors. A lot of ethical issues and especially for conditions where screening is not as easy, like Lefer-Almani syndrome, the germline p53 mutations. It's kind of requires total body screening, or else some kind of liquid biopsy screening. So in the past, people kind of debated whether what effect it would have on to tell people they have a cancer predisposition, but there's no intervention to
change your gene. You can't change your gene.
Yeah, change it.
But probably if I know the number, let's say your chance is just 5% more than something, and 5% more than something is nothing compared to your smoking habit, which raises 500% increase. So it's very important to tell probably the patient, so to get the information in a more precise way, just not scaring how many percentage extra. And that would probably help us, the ordinary physicians, to counsel to the patient.
Yeah, no, you're correct in that now oncologists might be the first to get the question, okay, what does this mean? And that's not their specialty. So it has created, some anxieties among oncologists, and try to become more educated about germline genetics.
Okay, lots of future opportunity and prospect of bright treatment effect will be coming on. And thank you very much for your beautiful future talk, or current talk, and it will lead to future happiness. Thank you very much for nice talk, Dr. Ladanyi.